Great. Thanks everyone for showing up today. I'm Mike Stadniski, the CEO of Photonics. Excited to talk about high parameter flow cytometry panel design today. Seddon is going to be our host. She earned her BA in chemistry from Williams College and her PhD in medical sciences, immunology, and virology from Harvard. She's got 20 years of experience in flow and immunology. And I warned her when she joined Photonics that it would be like being a kid in a candy store. And hopefully she'll be able to show you what that looks like today. She's published over 34 papers, received awards from AAI, the President's Rising Stars in Mucosal Immunology, and then multiple NIH Fair Awards. And so with that, I'll hand it over to Seddon, and then I'll be moderating as well. So please feel free to use either the chat, which we left on, or the Q&A function, which is at the bottom of your Zoom screen there, to ask questions at any time. So we like to keep it live, and so I will interrupt Seddon while she's presenting, and we'll go through that. So cool, let's go ahead, Seddon.
All right, thanks for the introduction, Mike. I'm gonna go ahead and start sharing my screen. Okay. Got a little. All right. Sorry, a little lag there. So one of the ways I think Mike has pointed out that Mario Roederer likes to think about flow cytometry is this idea of the number of colors doubling every 10 years. And so you can see, you know, on this graph, you have sort of the initial dyes and then sort of this build up to quantum dots with the brilliant violet, then substituting somewhat in there, the BUV, and now we have the NovaFluors. So we're really at an inversion now. Up until this point, the cytometers have been trying to match what's happening. But right now, dyes are really driving the hardware development. So you can see, you know, as we're talking about this, you have the FACS Vantage, LSR2, then the Symphony with convention.
Hey, so I don't want to interrupt you here. Can you put it on presentation mode? It seems to be lagging if you've done that.
Oh, okay. So what's happening? It's not, you're not seeing the slides, or it's...
We are. Yeah, can you try put it in presentation mode if you could, and then we'll see if it's lagging behind you. But you're, okay. Yeah, there you go.
Okay.
Yeah, try and reshare it maybe, and we'll.
Okay. Let me try to share it this way. Maybe that will be better. Is that better?
Let's take a look. I am seeing a big white screen. All right, try to put it in presentation mode, and let's see if that's better.
Okay.
Hey, there we go.
All right, good. Excellent.
Cool.
Sorry about that. So anyway, this is just showing, I guess, if you didn't see it before, I'll just skip quickly backwards so you can see, you know, that the NovaFluors are really, you know, building out from this place where you have, you know, initially some of these early dyes, then the BV and the BUV, and now we have the NovaFluors. So if you think about this, the dyes are now really, we're driving the hardware development. And so you have the LSR2, then the FACS Symphony, and building out to now we have spectral systems where we really just don't have the dyes to meet all of the detectors. So if you're thinking about 5 laser Aurora, there's 64 detectors, right now a max of 40 colors that have been tested, or even the Sony ID 7000 with 7 lasers and 188 detectors. I'm not sure if any of you guys were at the Bigfoot presentation, but looks like we now have a spectral sorter in the mix, so pretty exciting stuff as we're building out to more and more colors. So the title of the talk today is going to be focused on high parameter flow cytometry panel design with NovaFluors. And so there's this idea of really thinking about, we've had, you know, the dyes that we've had, and so we've designed panels around that. But what if you could really start over and think about, you know, what you'd want in a dye and what you're really not realizing you're having to design around. So this idea of spectral overlap, you've got these really bright dyes, but when they're taking up so much of the spectrum, it's really hard to, separate out your data at times as you build to more and more parameters. You're also thinking about antigen density, as well as the spectral spillover and spreading, laser cross excitation. So all of this affects what you're doing in terms of your panel design. So this is really based upon the performance of the dye, not so much what the cytometer can do. And so most instruments at this point are currently using up to these 5 lasers, so the UV, violet, blue, yellow, green, and red. Right now, there aren't any labels in the deep UV, and there's a zombie near in the IR. So there will be, of course, more dyes developed, but this is the spectrum that we're going to be focusing on at this point. And so one of the things we decided as we were looking at where to really design labels, what's happening with this blue and yellow green, and some of what happens there in terms of the dyes that are present have more to do with the fact that people were initially using this blue laser and the yellow green was added later, and it did make certain fluors brighter, but we didn't really stop to think about what does that mean in terms of it making those brighter. That has to do with where they're excited. And so what you end up having to deal with is a lot of spread in the system. So, you've got, for instance, where your primary detector is. This is some Aurora data showing you the UV, violet, blue, yellow, green, and red portions of the spectrum. And so in a conventional cytometer, your primary detector would be in this initial area that's marked with a box. However, you still have spread into the UV and violet from this particular dye. And if you're thinking about that even more so, you actually tend to have a lot of spread between the blue and the yellow green because a lot of labels are actually excited by both of these lasers. And so this is just showing an example of a few labels. So per CP, you can see there's spillover all over the place. BB515, you have the spill into the violet and UV, and BB700, even more spillover. So it's great to have a bright label, but you really want to have it where you want it to be and not somewhere else. So what if you could really design that fluorescent label from scratch? So how would that affect your panel design, to really step back and think about it? So that's what we tried to do. So in thinking about what, I, in terms of running my panel and really trying to look at my immunology would want, I want it to be bright and clean. So, I don't want to trade off brightness for being able to have a clean signal, because if I am, then I'm not able to look at a lot of other labels that are there. So you want it to be silent in the violet and to limit the cross excitation. Of course, you want the label to be consistent and stable. So, there are some tandems where there are issues with changes over time. So if you're doing a longitudinal study, that's obviously a huge issue. Or even, if you fix it and there's some changes related to the fixation, that can definitely be an issue. And then if you're really, getting to choose what you want to be able to control the brightness. So matching that, density of the antigen to how bright the signal is, and being able to turn that knob up or down. So that's what we really tried to do. And so this is the Phyton platform. And what you can see here, it was actually originally started in nanocomputing, and these formed these Phyton structures. And so it was actually this cruciform structure that's formed from single-stranded DNA oligos are then formed into this Phyton, which is this cruciform that you see here. And then basically you have a donor and acceptor label that are then placed on the single-stranded oligo. So that when it forms this Phyton, they're in a very fixed conformed formation. So why would you want to do that? Well, this allows you to take advantage of FRET, so the fluorescence energy transfer, forced resonance energy transfer. And so that allows you to tune the excitation and emission of the Phyton. And so then we can actually look at separating out, okay, where is the excitation happening and where is the emission? So really being able to change those dials and not have this sort of be a much more broadly excited dye, which causes a lot of the spillover from the cross excitation. Additionally, you want to be able to tune the brightness. So having the labels either on one arm or 2 or 4 arms, you can see here. And we can even do things like tunable signatures. And the nice thing about this platform is that we can then attach this to antibodies and use this to label, the antigen of your interest. So, sorry. So anyway, so this is just an example of the first 6 labels. And I do say first 6 because we will, be releasing more labels. Stay tuned. I think we have some exciting announcements coming at the end of June. But in terms of thinking about how this is labeled, the NovaFluors, the Nova Blue refers to the excitation from the blue laser. The Nova Yellow refers to excitation from the yellow laser. And then the naming is also 530 nanometers. So this is where the peak emission is, and 570 is where the peak emission is for this Nova Yellow 570. So it makes it really easy to understand, okay, how do I put this into my panel? How do I build out what I'm wanting to do? And one of the really interesting things that you see right away is, for instance, with the Nova Blue 610 and Nova Yellow 610, the fluorescence is actually similar in terms of the emission. So you see that it's emitting at a similar wavelength, but what's separated out is the excitation. So this is excited by the blue laser, this is excited by the yellow laser. So you tend not to realize that's a problem because a lot of cytometers have been built around this problem. Specifically, the Fortessa actually only has a trigon off of the blue laser. And this is because a lot of labels would be excited by both the blue and the yellow-green laser, but you could actually use more off of that, add an octagon or something like that, and really look at more labels even off that blue laser. So part of what people have done with designing flow cytometers is actually take this into account. So when you're looking to, at NovaFluors and want to see, okay, how can I incorporate them into my panel? We have several places where you can look. So FP-Base is a free resource. So the NovaFluors, you can see their excitation and emission. We're also now in FluoroFinder. So if you're looking to set up your conventional panel, you can actually very easily integrate the labels that you're already using with these and see how they're going to match up and where you might have areas of spillover or be eliminating those. Here, we're also listed in the Cytec full spectrum viewer. And one of the nice things that you can see when you look at the Cytec viewer is that our Nova Blue labels really are being excited off the blue laser, and our Nova Yellow labels are being excited off the yellow laser. So we really are separating that out. And so this brings me to sort of this comparison. So in this case, this is just showing some setup on the Attune or Fortessa and some of the suggested floors and what you could use in terms of photonics labels just on your conventional cytometer. And one of the first things that you see here, for instance, and this is what the Fortessa often has designed around by using just the trigon. So you'd have side scatter, FITC, and Percy-P that you often detect off the Fortessa. But PE actually would be detected off both the blue and the yellow laser. And you can see here, for instance, we have places, so Nova Blue 610 is most similar to PE Dazzle in terms of its emission, but you are able to change the filter and be able to detect that both off the blue and the yellow laser excitation here. There are also issues even with using things like Percy-P Sci 5.5 and PE Sci 5.5. There's still some overlap and spillover, even though these are separate labels that theoretically could be detected separately, but because of the spillover between the blue and the yellow laser excitation, you still have those issues. So to show you this in a little more detail, this is showing you, we have PE Dazzle, as I mentioned, and Nova Blue 610. And this is showing actually a plot of the absorbance or the basically where these labels would be excited. So you can see that PE Dazzle, similar to PE, can be excited by the 488, the 532, and the 561 lasers. Whereas Nova Blue 610, you can see really is just excited from the blue laser and very minimally off of anything with the yellow green. And then when you look at the Nova Yellow 610, you can see that there's almost no excitation off of the 488 laser, whereas it's primarily excited by the yellow-green laser. So again, separating out what you're seeing in terms of being able to look. So basically a 2-for-1, being able to stack these labels together. So in terms of panel design, this really means that you could go in and basically use these in place of current labels that you're using and be able to see more colors on your current instrument, whether that is a conventional cytometer or a spectral cytometer. And this is just an example of some compensation with the NovaFluors. So you can see here that, you know, I've mentioned that these labels are clean and bright, but you can see how well these separate in terms of looking at the compensation. You don't have some of this, you know, spillover where you might have, you know, some bleed over, which makes it hard to detect your signal, or broadening out of that in most of these cases. And in terms of, so this was done on the Attune. So I've shown you data now with the Attune. This is actually, this next data is done with the Symphony at the NIH. And you can see we basically looked at a number, Steve Perfetto actually at the NIH looked at a number of different labels, comparing either with the Nova Yellow 610 in CD4 or CD8 with the Nova Blue 610. And so this is just showing you the list of the comparators and the lasers off of that Symphony. And when you look at the Nova Blue 610, and right here, there's actually very few that are just going to be excited off of that blue laser. So you have the comparator, the Brilliant Blue 630. You can see that the Nova Blue 610 is not as bright as the Brilliant Blue 630, but it also doesn't have the problem of so much spillover across the spectrum. You can see that there's a large amount of spillover into the violet. And looking also at the Nova Yellow 610, you can see a similar picture. You've got comparisons with the Alexa Fluor 694, basically a PE dazzle dye, and ECD. So you can see that in these cases, you've got the Nova Yellow 610, which is either as bright or in some cases brighter, but doesn't have nearly the spillover issues, especially in the violet, that other labels do. And this is showing you, first off, the separation index. So when you think about brightness, what does that really mean? Well, brightness is really important in terms of being able to separate out your positive and negative populations. So one way to compare brightness between all these different labels that are detected, you know, off different lasers, et cetera, is to look at the separation index. And so you can see here that Nova Blue 610 and Nova Yellow 610 actually have a very similar separation index to APC. So they are still quite bright and usable dyes, but still not having that spillover that's going to complicate your analysis. And Steve pointed out that for the Nova Yellow, the overall spectral spillover sum for the CD4 Nova Yellow contribution into all the detectors was 8.58, which was lower than any of the other dyes measured on beads. So, you know, again, what does this mean for your data? Well, this means that it's going to be easier to interpret because you're going to be able to have a better separation in terms of your positive and negative signal. And also because you're looking, for instance, again, at the Nova Yellow 610 and Nova Blue 610, that actually you're not using up 2 of your labels. So the PE dazzle, for instance, as I've already mentioned, would actually take up both of these lasers. So you also have less spillover in terms of cross excitation. So in this last slide, this is the idea of upgrading a panel. So this is taking the filter set that's on the Attune. It's slightly modified in terms of switching out this BL2 filter, but everything else is similar. And what you can see here, if you wanted to run, say, a memory T-cell panel, you wouldn't be able to do what I've proposed here, because you have PE dazzle being detected off the BL2 and the YL2. So this would be, sort of not possible to run this extra PD1. you'd also have the conflict between CCR7 and CD28 because of the per-CP Cy5.5, PE Cy5.5 spillover. And so this is just showing an example. Actually, I'm in process of doing this experiment. We, unfortunately, with the COVID situation, have had some access issues and are getting back into that, just probably like a lot of you have had in the process. But this is basically, I'm showing you the setup for that experiment, which is that we can actually run the NovaFluors off the blue and the yellow lasers, and you get these two extra labels that you can look at. For instance, if you're trying to look at memory T cells, PD1 and CD28 in this setup, and using the exact same instrument that you have, you know, the Fortessa, you could switch out depending, just because of the trigon, the limitation, but you actually could eventually get an octagon or something like a Fortessa. So this is just showing one way that you could really upgrade a panel. And you could do this with whatever instrument that you have, whether that is, you know, an Attune, Fortessa, Aurora, Symphony, etc. So I don't know if there are any questions this far. All right.
Helps if I unmute. Yeah. Well, yeah. So I've got a question here. So will you be speaking to you know, its use in intracellular staining?
We are just about to do that experiment. I'm really excited. So we are not going to be showing that here, but that is literally an experiment that's planned for the next, I would say, 2 weeks. So. And then,
yeah, and the next question is also kind of leading down this the high parameter route, which is, you know, can you describe any work that you've done, either, you know, yourself or with partners, in working on panels for the FACS Symphony?
Yes, yes. I've actually been working with a partner recently who wanted to upgrade his Symphony panel, and was showing him actually that this could be done. So one of the really nice things about, I'll just show you this on for instance, this proposed Attune panel, but again, the Nova Blue 610 and Nova Yellow 610 means you can separate out. So, you know, he's able to basically get a 2-for-1 here. So taking out the PE Dazzle and substituting in 2 of the NovaFluors, and then also looking at, for instance, PE Cy5, substituting in Nova Blue 660 and Nova Yellow 660. So again, this 2-for-1 stacking of the labels. So that's specifically on the Symphony. In this case, Nova Blue 530 is most similar to FITSI, and Nova Yellow 570 is most similar to PE. And so this particular client, I guess, customer, he was interested in actually using GFP and tomatoes. So he, you know, didn't want to give up too many of those labels there, but actually was able to get, you know, 2 additional labels even into his Symphony panel.
Great. Yeah, and the next question here, and it might be worth going back to the slide off of the X50.
Mm-hmm.
Was the 532 or a 561 laser used there?
It is in this case, the 532.
Go back, yeah.
Yeah. So I believe if I go backwards, you can see, going back a little further, you can actually see that you're even going to get better brightness or separation with excitation from the 561.
Yeah, exactly. So it's a great question, because yeah, I mean, you're seeing pretty good, you're just seeing really good performance, and you're seeing good spread performance, for instance, off of, you know, both these labels, even when there is a 532 present. And so, you know, really good performance off of 561. And actually, just out of curiosity, some of the stuff that we're looking at off the Attune, what does that have?
I believe the Attune has a 561.
I think so too. So yeah, it's true. The 532 actually would essentially cause some more trouble, if you will, yeah, for these labels, but we're not seeing that. So it's a great question.
Yeah. All right. Anything else? All right.
Oh, keep going.
Okay. So the next thing I wanted to talk about is the consistency and stability data, which is, I think, really exciting, especially for those of us who have dealt with tandems, or in this case, I like to refer to this as the absent-minded graduate student experiment, because I have been in labs with someone that will forget, and you find the antibody floating in, you know, a pool of what was ice and now is water. So what does happen if you leave our labels out? And what you can see here, we've actually taken this out to 3 weeks, and you can see that there's no change in the fluorescence if you just leave this on the bench for, you know, this data is up to 14 days, but we have done this up to 3 weeks. So incredible stability. And this is really because of the DNA structure that really acts, I think, as like a solubility cage. And so you can, although you get the benefits of the tandem, you don't have the problems that you would have with the tandem, because it's protected, so you're not losing part of that signal and having that change over time. So you don't have to, you know, for instance, for PE-Cy7, people are familiar with the idea that you need to run the PE-Cy7 off of that antibody to make sure that that lot is, you know, what you're compensating off of, whether that's for spectral or conventional. And so this is showing stability data after fixation. So initially here you either have unstained or the Nova Yellow 610. This is actually a 1 cluster label with CD3. And so you can see the separation here. But if you leave this then fixed, I think Mike likes to refer to this as the COVID experiment, which is a good note. And we certainly have, you know, again, had sometimes accessibility issues in terms of instruments. And what you can see here is there's really no change. These cells were fixed in, I believe, 2% paraformaldehyde. We've also done the test with 1% formalin, and there's really no change in that brightness. So it's really nice to see that, you know, this is, again, leaving this in the fixation, not washing it out and waiting. So. And this slide also, in terms of consistency, what's strange is the way to really best explain this slide is to draw your attention to the more of the issues than the other. Each of these are actually showing 2 lines. You can barely see that there are 2 lines here, but so you can see this most in this Nova Blue 530, and this is still less than 5% change in terms of the fluorescence from batch to batch. So these are incredibly stable, incredibly consistent as we prepare new batches of NovaFluors. So the last of these knobs was looking at controllable brightness. So, you know, we've looked at how bright and clean the labels are, consistent and stable, so the brightness. So the knob on this is, as I mentioned, this 1 cluster, 2 cluster, 4 clusters. And so what we refer to as these clusters are the different arms of the phyton. So you can actually build out this brightness. And this first slide is just showing that the networks can be replicated. So there are actually slight changes you can imagine in the geometry of the phyton. We show it in this sort of 2-dimensional cross structure. But of course there is, you know, a three-dimensional space with the DNA. And so but even if you do this on one arm versus, say, arm 1 versus arm 3, you can see that the network is replicated and you have very similar brightness there. And so then you can actually take from 1 cluster to 2 clusters and actually double the brightness. And of course, the question is, that's nice, you can show it in terms of the floor, but what happens with, you know, my panel? So in this case, I'm showing the Nova Blue 610. And you've got either 1 cluster here or 2 clusters here. And this is just showing label only, so very low background. And then you have staining for CD3, either the 1 cluster or the 2 cluster. So as you would expect, you see similar percentages of cells, but the brightness per cell is actually doubled. And so you can see that that really then helps you look, if you're looking at a very low signature, you know, that really is important. Most of our labels, the labels that we actually sell are the 2 cluster version. You can actually build out to the 4 cluster version. And so that's what I'm showing here. It's just the brightness again. If you take this from 2 clusters to 4 clusters, again, you have almost a twofold increase in brightness. And this is just showing that if you normalize those results, it's very, very similar in terms of its performance. There's a little bit less spillover here, and this is just because of the change in conformation. But you can see that this is actually very reproducible even as you build out to more clusters. So this is then looking at a 2-cluster and 4-cluster phyton. And this is the Nova Yellow 610. And as I've mentioned, this is most similar to PE Dazzle, except that, again, PE Dazzle would take up both the Nova Blue 610 and the Nova Yellow 610. And by dividing it out, you have that really nice bright signal, but actually a 2-for-1 in terms of what you've been currently using with your cytometer. So what you can see here is we've got the 2-cluster versus 4-cluster. Again, the MFI, you have nearly this doubling here, and again, log scale, so you can see that. And then also on this same plot is PE. And you can see PE is incredibly bright. However, it's spilling over both into the yellow 2 detector, so you're kind of losing some of your signal that you're able to see there, as well as the cross excitation into the BL2. You can see that for the Nova Yellow 610, the spillover is very much minimized. So you really get, you know, a similar type of brightness, but much lower, you know, cross excitation and spillover. So we also have, I've mentioned about the 4-cluster Nova Yellow 610. Currently that is actually in beta testing. And there are a number of applications where this could be important. So looking at, say, small particles and extracellular vesicles, we have some partners that are interested in that. That also, you know, as I mentioned, PE and PE tandem replacements for looking at nearly undetectable drug targets. And looking as well as checkpoint inhibitors and the ACE2 detection. So there are a number of applications, and if you're interested in building out to that brightness and want to talk to us about the 4-cluster, please do. So I don't know if this is a good place for me to pause for any other questions.
Yeah, let's go ahead and do that.
Okay.
So question here, can you combine phyton NovaFluor conjugated antibodies with a cocktail of oligo-conjugated antibodies used for like CytSeq or ReapSeq? For example, you know, the total-seq or AbSeq antibodies. And then will it impact downstream applications such as sequencing?
It's a good question. So, you know, basically I'll show you how we actually do our conjugation, but again, it is using an oligo label. And then, you know, there's basically a 3-step process where you get rid of the excess oligo label. And then you do a slow annealing process from 50 degrees down to 4 degrees. So again, once these phytons are on there, you really shouldn't have any problems in terms of that. And also these other labels would have, I believe, you know, also different sequences, so that you really shouldn't have any issues in terms of the, you know, sorting or your downstream applications. We haven't tested all of that specifically, but there aren't any reasons that I could come up with that there would be an issue.
Yeah, and it's worth noting that, you know, because of the efficiency of the oligo, right, you can essentially add the labels. We do actually have a modality where you can add the labels directly to some of these other total-seq or AbSeq antibodies, right? And so that is something that we've certainly tested, that conjugation. So you can imagine combined workflows where you're actually adding the phytons directly and then using them in that application. So I think particularly if that's of interest, you know, I'll reach out to you, because I think it's a very interesting thing that I'd say we're actively looking at to make even easier than now. Mm-hmm. Yeah, another question. Similarly, as the Nova dyes are oligo-based, do you notice any increased background when staining intracellular or nuclear targets?
Well, again, I'm actively doing that in the next 2 weeks. So I promise I will have that data for you soon. I don't, you know, think that there will be any, but we have to actually test it in an experiment. So definitely to come, almost there. I'm dying to do the experiment. Mike can attest to this. So we've just had some delays in terms of instrument accessibility, which is opening up as the country starts to open up again.
Yeah, I'd add to that we've designed for the conjugation kits that we're using for this. We've designed those sequences to make sure that they don't bind anything intracellularly, right? So we've been very attentive to that to make sure that we can expand out what's being done there. And so we're starting with transcription factors and then trying to move really out from that. And so I'd also say phosphostats is something that we're very interested in partnering on. And so that's another area where we don't have very deep expertise there. And so really happy to discuss that. And if that's an application that's of interest, be great to partner up.
Yeah, absolutely. All right. So now I'm going to talk about a truly extreme high parameter panel. This was work that, as we were thinking about, okay, where do we, really test out the fluors? We've talked about how this works. So I felt like one of the best places to start would be the panel that Cytec had already set up. So in their case, it was a 35 color panel. One of these is not commercially available. So starting with a 34 color backbone and then testing out the NovaFluors. And part of the question that I had is, if we're just dropping these into a panel, are there any issues without looking at, the panel design? These should be unique. So what do we see in terms of what that does to the fluorescence in the panel? So building from 34 then to 40. And you can see here, this is just showing the panel, again, as I said, that Cytec had put together, and then literally just sort of trying to be agnostic and dropping in our labels. So we have the Nova Blue labels here and the Nova Yellow labels here, and you can see where their primary detector is predicted to be. Also, tried to use both chemokine receptors as well as other labels that would be on, for instance, CD19 was one of the ones we eliminated, but I didn't just try to use just bright markers to illustrate this point, to really test out what we're seeing. So this is, what looks like a bit of a messy spectrum, but this is, if you put all of the, fluors in there, the 34 color panel plus the NovaFluors, you can see that. And one of the things I really like seeing is, for instance, the uniqueness of this Nova Blue 610. When people are talking to me about panel design, it's one of the places where you can actually see there's almost like a hole waiting for something to be built out there. You can see here also, you've got the, Nova Blue 530, which is similar to some of the BB 515 and FITSI, as well as the Nova Yellow labels. And so I'll get more into the details of that, but just showing you that overall matrix. So this is the similarity matrix. And what you can see here when you look at the 40 color panel is that there are, and then group it in terms of this, you can see that the violet dyes, there are actually some areas where there's already some bunching in terms of the violet range. And so one of the things with just the 34 color panel is that there was already some spillover into the violet. So being able to know that we're not contributing a lot to the violet is very helpful in terms of these labels. Also, the BB 515 and FITSI unmixing is challenging. So the Nova Blue 530 actually makes this worse. So in general, I would recommend using Nova Blue 530 with either BB 515 or FITSI alone, because this actually introduces less complexity into your panel design. So FITSI and BB 515 are basically have a similarity index of 0.98. And actually Nova Blue 530 is less similar to either of those. So if you use one or the other with the Nova Blue 530, it actually makes the unmixing a bit easier. You also have areas with PE and PE dazzle. So the Nova Yellow 570 is most similar to the PE-CF568. The PE-Dazzle, again, Nova Blue 610 and Nova Yellow 610. And PE-Sci5 is most similar to the Nova Yellow 660, and as well as the Nova Blue 660, really. So as you're looking ahead, I want to just start by drawing your attention to the spectral spread matrix of the 34 color backbone. So again, you already see that spillover into the violet. As I already mentioned, that was going to be an issue. And again, BB515 and FITC already have some bigger contributions here. So the test I was really interested in was understanding what do each of our labels contribute. If you drop them in one at a time, what does that tell you about panel design? Because to me, that was one of the mysteries sort of going into this is, you know, how are these going to specifically affect some region? Is it going to affect everything throughout the spectrum? So trying to look at this delta SSM comparisons. So you can see, you know, on the previous slide, you know, there are areas. So this is just showing how much the labels change that. And again, as already mentioned, the Nova Blue 530 does increase this spectral spillover, but really just at the place that you'd expect to see it. So 515 and FITC, and you can see this is on a bigger scale. So the easiest recommendation is that I would use Nova Blue 530 with either one of these in a spectral panel. In a conventional panel, you know, would choose one of these three, but you can actually distinguish them in a spectral panel. One of the cool things about Nova Blue 610 and 660 is there's actually little to no change induced by adding these into the panel. And you could see that based on sort of that really sort of slightly more open area in terms of the blue laser. So then looking at Nova Yellow 570, 610, and 660, again, with this idea of plug and play, you can see that there are small changes in the spillover. Again, this is, you know, that smaller scale in terms of the change, but the Nova Yellow 570 affects exactly where you'd expect to see it and nowhere else. So PE and CF568. The Nova Yellow 610, again, it changes this for PE Dazzle, and the Nova Yellow 660 for PE Sci5. And so it's really very plug and play in terms of looking at the data. So, you know, what we've done is added 6 colors with marginal impact on the spread, except for this already difficult BB515 and FITC combo. If you look at the complexity index, I think it's a 9 point something just to have those 2 colors together. So, you know, generally I would recommend around that anyway, but it is does add less complexity to add one or the other with Nova Blue 530. Additionally, there's near zero spread added to the other channels. So this then, what does this mean for your data? Well, it leads to easier experimental design, higher resolution of your biological populations, and easier data analysis. So
maybe I'll interrupt you there because I think, and I believe this was in reference to the Cytometry spectral plot, if I'm not mistaken. Mm-hmm. And hopefully we addressed part of it, at least with some of the spread data, was, you know, what are you seeing cross-excitation problems, especially in the 660 emission range?
Yeah, so we can go back. So you can see, so the red is turned up a little higher here. So you can see that just for the Nova Yellow 660, you really don't see this for the Nova Blue 660. There is a small increase also for APC and the APC R700, but very, very low there. So really still very low spillover.
Yeah, so there's probably 2 additional items to add there, right? Which are APDs are very sensitive in the red, and the gain on these is turned up very, very high to provide that separation of, what is it, APC 700 and Alexa 647, is that right?
Yes.
And so you're seeing that, but I think more, I guess maybe perhaps more impactful is the point you're making, right? Which is that despite that, you don't see really much in the way of spread contribution, even when you've got, you know, these other labels in that 660 range. So hopefully addressing that question, because yeah, you certainly see it on the spectral plots, but then I think the spread is really what you'd like to be able to see here and say, okay, is there actually any contribution to the data analysis?
Yeah.
So hopefully answer that question. So.
Yeah, no, that's great.
Cool.
Are there any other questions or? All right. So this is just showing you, this really cool subway plot that Mike has put together just for visualizing the data. So again, taking this 40 color panel and then how do you really look at, okay, illustrating, how you're doing your gating and separating out the populations and illustrating this really complex data set. And so you can see here, this is showing you, the individual gating, sort of the nodes at these points, as well as the expression of different markers throughout these different subpopulations. And, one of the things, for instance, there is, this population of CD20 negative, IgD positive, which really comes from this spillover in the violet. So this is, gated out. And if I wasn't, showing you the individual gating, you can, get around some of these things, but we're trying to be really transparent with the data and show you, okay, where are the issues? And so some of this extra spillover in the violet does make some of that analysis a little more challenging. But in terms of your biology, you're still really able to resolve a huge number of populations. And this is a t-SNE plot showing that, while there are, identified populations, there are also other populations that you can actually use something like this 40 color panel to really look deeper and discover new populations in your biology. So we have made this data available. So you can either get it, we have the Above and Beyond 40 Colors white paper at phytonics.com. We also have made the data available at our GitHub link, as well as Flow repository. So if you want to look at the data, I would say don't take our word on it. Feel free to dive in, and hopefully this helps you as you're looking to design your own panels. We want to enable you to look at the biology that you want to look at. And so that's really why we've tried to make this as a publicly available data set. So I don't know if there are any additional questions.
Nope.
Okay. So then how do we do this? So right now, in terms of being able to test this on your instrument and say, okay, if I drop in these labels, in terms of my panel, how can I test this out? Well, we're offering a human and mouse CD4 Nova kit. So this has these first 6 NovaFluor labels. So the Nova Blue 530, 610, 660, as well as the Nova Yellow 570, 610, and 660. And what you can see is these are competitively priced. And so you're able to really get at what you want to test out in your panel without having to share that. I will say that if you are interested, I'm happy to just, I've actually had some people where they just share what fluors and what instrument. And so I'm happy to provide any advice if you need that, but this makes you able to really test this out for your own system. We also are offering the Nova block, and this is really has been important in terms of some of the PE and APC tandems use a lot of these side dyes, and there's non-specific binding of the NovaFluors, as well as some of these PE and APC tandems for macrophages and monocytes. And so you can actually use this blocking solution and actually use this, and it works well with a number of other blocking reagents that I've tested them with. We'll have that a white paper on that soon, showing you all of the comparisons. But yes, that's actually very helpful, and it only takes 5 minutes of blocking. So you can mix it in with, say, we recently did this with some of the mouse CD4 using mouse serum, rat serum, FC block, Nova block, and all of that together, and there's no issues. So it's a very short blocking step. Additionally, with being able to label these, so we have a conjugation kit that we're currently offering. And these are all in a 1 by 100 microgram antibody setup. And so you can actually purchase them individually or as a 6 by 100 kit to look at all of them in your panel. And so this allows you to look at, for instance, your specific antibody of interest in there. And I'm just going to go into a little more detail on the conjugation kit for those that may have questions. You basically, in this case, we have a single-stranded DNA linker, and it is, we recommend the antibody be at 1 mg per mil. We also recommend that you not have any other proteins there, because otherwise you're going to be labeling your other proteins. It's just that simple. It's not that you couldn't use it, but you would just be labeling some background and losing some signal. So the idea is there's 3 pipetting steps in this initial part of the process. And basically, you just let this go overnight at room temperature. The second step is to remove the excess single-stranded DNA linker. So it's basically 2 precipitation steps in an Eppendorf tube. And then this last step is similar for anyone that's done PCR. You're basically mixing the antibody with the labeled antibody with the phyton, which also has a complementary single-stranded linker, and basically slowly cooling from 50 degrees down to 4 degrees. And then your NovaFluor antibody is ready to use. So it's really straightforward in terms of that. And this is just a slide showing you all the buffers, but really the main thing is that you want the antibody concentration to be at least 1 mg per mil. That's just because you get a better conjugation in that in terms of your labeling. And again, you really don't want excess protein there. For folks that are interested in higher custom conjugation, we are offering to do the conjugation in-house if you have 1 milligram or more of antibody. And so feel free to ask us about that if that's something that you're interested in. But you can see that, all of these are competitively priced to some of the antibodies you're already using. So, there's a very minimal step to get this into your hands so that you're able to use it. So, what does this really mean in terms of your data? That's really the important part. So, our labels are bright and clean. We've limited cross excitation. So this means you're getting the best, performing fluorescence for, with clean data. So you're really not getting so much of the spillover that you are accounting for whether or not you're, paying attention to that. The labels are consistent and stable, so you can leave them on a bench or in fixative. The, excitation and emission, we've made that tunable. So obviously that's something that we're doing as you buy the floors, but we will be moving more dyes into the future. So that's really exciting. This truly can be used as plug and play. I mentioned for, as an example, even with a conventional cytometer setup, you can substitute in, for instance, PE Dazzle with, separate into the 2, the Nova Yellow 610 and the Nova Blue 610, or with something like PE Sci 5 or PerCP Sci 5.5. You can use the Nova Blue 660 or a Nova Yellow 660 again to separate out those 2 signals. So again, this 2-for-1 sort of stacking setup. And this, what does this do in terms of your panel is hopefully will allow you to discover more. So you can stop letting dye performance get in the way of your panel and get higher resolution on the data you're interested in. So with that, I'll wrap up the talk. I've got my email address listed there as well as Mike's. If it's easier to remember, info@phytonics.com is also a great place. So if you want to reach out, we're happy to answer any questions, or if you have any, ideas or, anything you're interested in investigating, we're there and happy to answer that.
Great. We've got 2 questions here. Hopefully I'm not breaking up too bad.
No.
Okay. First one is, have you tried them in sorting?
We have not, but That's only because we were actually just literally limited in terms of instrument access right now. So I think as things get going, that's certainly something that we'll be doing soon.
And the second question, maybe my other favorite question after the genomics one, is what and when are you going to be releasing new colors?
Yes. So I think we're going to be making an announcement. It's actually listed on our web page, as well as the email that you get out. So look towards the end of June, we'll be making some exciting announcements.
Yeah, it's June 24 and 25. Sorry, I was just looking.
Yeah, I can remember the exact dates. So.
Great. Okay, well, we'll hang out for a couple minutes for any questions. Okay, great. Cool. Yeah, so we'll hang out for any more questions, and happy to address those in the Q&A or in the box here. I feel like we need outro music. Here we go. Cool. All right.
Yeah, that's the sad part of missing Cyto, guys. Mike was organizing a fun run of the music festival.
No.
I was kind of excited for that myself. So.
No music festival, Ron. I can't coat myself in black light paint and run through Philadelphia.
I was excited for that.
I know you can't do that virtually, it turns out.
Yeah. Well, I mean, I guess you could, but it's not nearly as exciting.
Yeah, I do think my neighbors would be worried.
For the GoPro.
Yeah, exactly. Yeah. Great. Cool. We'll go ahead and wrap this up. Thank you so much for the folks who attended. Thank you, Sid. That's great.
Yeah, thanks so much. I really appreciate it.